Alright, today's video is on mitosis. Why is mitosis important, you say? Well, it's something that's going on in your body all the time. You started out as this nice, cute, teeny, tiny little embryo here, and it is going through the process in order to get where you are today. It had to go through mitosis over and over and over again in each and every one of your cells. Another living thing, this little starfish over here, if it lost one of its limbs, it can regenerate them through the process of mitosis. Let's say you fell and scraped your knee. Well, the scrape has got to be repaired somehow, and you got to thank that process of mitosis because it's going to repair and replace old worn-out cells and ones that have been damaged. What are the objectives of this video, you say? Well, by the end, you'll be able to explain what the purpose of mitosis is, identify the types of cells that go through mitosis, identify the stages of mitosis, prophase, metaphase, anaphase, and telophase, explain the purpose of cytokinesis, and differentiate between somatic cells and gametes. Mitosis is by definition the process of nuclear division, where an exact copy of cells are made, and these cells are genetically identical. So if you look at a cell, for example, the one down here in this blue circle, it has 46 chromosomes. By the end of mitosis, it's going to have two cells that each have 46. So there are two identical copies of the original one. And that's mitosis in a nutshell. We're going to create two identical cells. Remember, mitosis follows interphase in the life cycle of a cell. It's going to be after the second growth phase, and it's going to run through PMAT, we call it. Prophase, metaphase, anaphase, and telophase. In this diagram here, we're showing a prometaphase. That's nothing you need to concern yourself, so just ignore that one. And then it's followed by cytokinesis. When we talk about a process of mitosis, we have to talk about two new vocab words. The first one is diploid. And diploid is going to be referring to the number of sets of chromosomes that are inside the particular cell we're talking about. So we're talking about mitosis, so we're usually going to be talking about a diploid cell. A diploid cell is a cell that has two complete sets of chromosomes. So if you look at this diagram here, I have three pairs of chromosomes. They each have an exact copy of itself. This one has two, this one has two, and this one has two. You get one of these from your mom, one of these from your dad. So it's a diploid cell. It's referred to as a 2N. The other type of cell that exists is a haploid cell. Let's think of half. It has half the number, only one set of each of the chromosomes. It's represented by a little n. So it has one chromosome here, one chromosome here, and one chromosome here. Very important. You want to add those into your notes. So we're working again in mitosis mainly with diploid cells. Where in the heck do haploid cells exist? They're going to exist in this little sperm and egg over here, which is where you came from. So your dad's sperm had 23 chromosomes, your mom's egg had 23 chromosomes, and they mix to form 46 chromosomes, and that becomes the diploid cell, because you'll get one chromosome from your mom, one chromosome from your dad. If we wanted to look at a picture of your chromosomes, we have it right here in front of you. That's a karyotype. A karyotype is just a picture of someone's chromosomes laid out by size and shape in organized fashion. So you'll notice in this karyotype we have all of the chromosomes numbered 1 through 22. The last pair, the 23rd pair, is your sex chromosomes. They're all arranged by height or length. In other words, the tallest ones come first, the shortest ones come last. With the exception here of your sex chromosomes, if you're a female, you're going to have two X's. If you're a male, you're going to have an X and a Y. But you notice the difference between the X and the Y chromosomes. The Y is a much smaller chromosome than the X. You notice these all have these black bands on them. Those black bands are a stain that indicates where a certain gene is. And a gene is a particular, is a particular location on a chromosome that codes for a trait. So if I look at chromosome number 9 up here on the top, this could be the space or the gene that codes for eye color. Or over here on chromosome number 3, this dark spot might be the gene that codes for your eye color. So all of these darker areas are genes, and they all code for a different trait, whether it's eye color, hair color, height, weight, muscle structure, and so forth. So another one of the vocab words we kind of want to make sure you have in your notes is gametes. Gametes are what we get, general term we refer to as your sex cells. All living organisms, or most living organisms, have gametes. And if we refer to the sperm, that would be a male sex cell, and eggs are female sex cells. Somatic cells are what we call all the other cells in your body except the sex cells. So your liver cells, your skin cells, your muscle cells, we could call those somatic cells. But your sperm and your egg, those are gametes. And if we wanted to just refresh our memory on all the silly little vocab words we have for DNA, between chromatin, chromatids, remember that DNA is the strand of nucleotides all strung together. That's a sugar, phosphate, and a nitrogen base. And when they wrap themselves all up, they can, with a protein, they're called chromatin. And when they're tightly coiled, we call them chromatids. And this down here would be a nice picture of a replicated pair of sister chromatids that are held together by their centromere. So what is the purpose of mitosis? Well, the purpose of mitosis is to allow for growth and regeneration of cells. So you take this little tall man here, he's gone through lots and lots of mitosis to get the height that he is because he's increased the number of cells he has in his body. The little lizards over here have gone through regeneration because they've lost a part and they've gone through mitosis to bring it back again. So to review, what types of cells undergo mitosis? All the cells in your body, with the exception of your sex cells. So that would be all somatic cells undergo mitosis. Sex cells are going to undergo a new process that we're going to learn next. So just to refresh your memory again, if we look at the cell cycle, again, we started with interphase, where you spend 90% of your time. The cell still grows right after division. Then it goes into the S phase, where the DNA is makes a copy of itself. You can think of it as the S or the sister chromatid phase. Then it grows some more in anticipation of mitosis. That's where we're going to take all those sister chromatids and divide them up equally to either side of the cell, so then we can split them in half and have two identical cells. So G1 is for growth. S is where DNA makes a copy of itself. You go from, in humans, for example, you go from 46 chromosomes to 92. Then you have G2, which is more growth. Then you have mitosis, which is the nuclear material or the separation of our chromosomes. We're going to take those 92 chromosomes and put them into two cells of 46. Your end result, star this is the most important part, you end up with two genetically identical cells. If you don't have that now, write it down again. Mitosis results in two genetically identical cells. Very important. So let's look at a picture of that. Here's a cell with one chromosome. This would be in the G1 phase. It goes into the S phase and it makes an exact copy of itself, where it has a sister chromatid. Then it goes into mitosis and cytokinesis, where those sister chromatids are separated out across the pole of the cell, and then eventually into two individual cells. So we started with one cell, we end with two cells that have one chromosome each. So that we've created identical cells. In humans, we would start with 46 chromosomes, go to 92, and then two cells that have 46. Other organisms, for example, if we were to take an ant, ants only have two chromosomes, and they're going through mitosis all the time, just like you are, growing and repairing their cells. So they have, in one of their cells, two chromosomes. They go through mitosis. They end up with two daughter cells, each with two chromosomes. A pea plant, they have 14 chromosomes. So when they go through mitosis, they produce two cells that have 14 chromosomes each. Dogs have 38 chromosomes. So when they go through mitosis, they create two new cells that have 38 chromosomes each. You get the idea. Mitosis is creating two cells that are genetically identical. We call them usually daughter cells just to signify a new generation exists. What are the phases of mitosis? First you have prophase, and that's what pro means, first, and that's where we have the chromosomes are condensing down so they become really visible. The nuclear membrane starts to break down so you don't see it anymore, and spindle fibers start to appear. Prophase is followed by metaphase, which I think is like meeting in the middle, which your sister chromatids are all lined up in the middle so they can nice and neatly be separated apart to ensure that the correct number of chromosomes is going to end up in each individual cell. Followed by anaphase. Ana, to me, it's like they're apart, they're moving apart, and that's when the spindle fibers pull down on the centrioles there, and they're going to pull these sister chromatids apart again, so we have equal separation so that each new cell ends up with exactly the same as the parent cell. Then we have telophase. I think of it's the end, so it's just about the end of the process, and here we have the nuclear membrane starting to reform. The chromatids, the sister chromatids, are starting to unwind, so it's turning into chromatin now, which is the uncoiled DNA, and you start to see the pinching in here. And if this is an animal cell, that pinching in, remember, would be called a cleavage furlough. So then we're just about ready for cytokinesis, and cytokinesis in animal cells is predeceased, or you start with the cleavage furrow, which is the pinching in. In plant cells, you start with these little vesicles of cellulose that are going to line up and eventually fuse, and those are going to turn into a cell plate and then eventually the cell wall. And that's all there really is to the whole mitosis. But if you wanted to get a little bit of practice, let's look at this slide. We have lots of individual cells here. Most of them are in interphase, and because remember, the cells spend 90% of their time in interphase. This one, the chromatids are nicely separated, so I would give that a telophase. This is nice anaphase. Here's a nice metaphase. This is late interphase, early prophase. So hopefully you can go around here and kind of get used to looking at them. The last thing I have here in this presentation is a list of your vocabulary words that you really, really need to know. And there's an awful lot of them, so you have to spend a fair amount of time getting a good handle on them. Excuse me.